I got my PhD from Don Hunt, and when I told Don Hunt that I'm giving a talk here, I told you already you should invite Don. So Don was nice enough to send me a couple of his historic slides that I'm using in this talk. But I'm also going to discuss the project that the previous speaker already mentioned, this paper I'm also talk about. So these are all mass spec-centric papers that are talking about detecting HLA peptides. So Don really pioneered this field, and recently everyone is working on those. And so this talk will be very technical, and I hope I can try to tell you why is it hard, and I agree with the previous speaker that detecting these peptides by mass spec might not be as straightforward as it seems. So that was actually the perfect introduction. So while everyone else is, positive, I'm like more giving you the reason why things might fail. And because I will be talking about mass spectrometry, I would just like to spend 3-4 minutes to very briefly go over how we sequence proteins and peptides actually by mass spectrometry. So usually what we take a protein, much like the immune system, in order to detect it or use it for anything. As mass spectrometrists, for the most cases, we have to chop it up into peptides, because peptides are easier to separate and peptides are easy to sequence. Intact protein mixtures, not so much. But once you have a peptide mixture, might it be based on enzymes that we use to cleave those peptides, or might it be that the proteasome generated peptides. These peptide mixtures are then injected with HPLC into a mass spectrometer, and we get 3 sets of data. The first one is a base peak chromatogram. You see time here, and you see the most abundant peptide at a given time and that elution profile. So that might not tell you much more, but then a little bit of the complexity and maybe how much peptides you have. But if we take a snapshot in the mass spec at this given time, we see what we call a single stage, a full scan, or an MS1 scan, where we simply measure the masses of the peptide that we are observing that reach the mass spectrometer at the same time. And even though we call it mass spectrometry, we're really measuring mass to charge ratio. So something that doesn't carry a charge in positive electrospray is very hydrophobic or negative. We won't see it. So and if you see a peptide that weighs 1,000 and it has 2 charges, you see it at 500. So and you can see that this is a single mass spectrum, but you can easily see that there are 100 different peptides present at a given time. So we inject them with an HPLC. So the HPLC is maybe 20, 30 seconds, the peak is wide. And your peptide that you're interested in is most definitely going to be right here, that the previous speaker said, the abundance bias. If you're a peptide that is here, you always get picked for sequencing. If you're down here, it might be very difficult. So in this, again, it just gives us a peptide mass to get the peptide sequence. We start usually by taking the most abundant ion, kick everything else out of the mass spectrometer, and then we're breaking this peptide apart, and then we record what we call an MS-MS, an MS2, or a tandem mass spectrum. And this is a fragmentation pattern of this peptide, and where we cleave along the peptide backbone. And in 99.9% of cases, these spectra are submitted to a database search. And in very few cases, they have to be sequenced by hand. And my focus area is actually in trying to come up with ways to interpreting the sequence out of those spectra when the traditional ways really fail. So, and I'm not going to show you really how to de novo sequence. I just want to give you a very brief overview of how we actually get the sequence. So what you see here is this following peptide right here, and this is a corresponding peptide spectrum. So we have a certain ion series that comes from the N-terminus. That means the peptide breaks apart here. And remember, is mass to charge ratio. If the charge retains on the original N-terminal piece, these are called either B or C ions, depending what fragmentation method you use. And then we also have a C-terminal ion series that we call either Y or Z ion. And what mass spectrometrists like to use to digest their protein mixture is trypsin, because it generates an arginine or a lysine. And then you can very easily see that these can be protonated, so you have a chance to actually see these fragment ions. If you look at HLA peptides, nature's digested for us, so we no longer can direct what we want to have at the C-terminus. And if there is no basic residue, it might be very difficult for us to detect. So, and then you have this peptide right here. Let's say this is a doubly charged peptide. Another reason why we like trypsin, because the N-terminus usually carries charge, and in trypsin case, also the C-terminus. Again, HLA peptides might not be the case. So, and if this had 2 charges to begin with, and it breaks apart here, 1 charge remains on the N-terminus, 1 charge remains on the C-terminus, then we observe 2 fragment ions. That, of course, again, doesn't give us a sequence, but if all of the peptide bonds are cleavable more or less equally, we should see a random distribution, which again is not necessarily always the case, but otherwise proteomics wouldn't work. So for the most cases, it has to be. What you see here is now that the N-terminal piece, if it breaks one residue over, it increases in mass by 113, which is the mass of leucine or isoleucine. At the same time, the C-terminal part, this peptide here, now got truncated by the same mass, and it's decreasing by 113, and so on and so forth. The next one is 129, that's the mass of glutamic acid, and so on and so forth. And of course, nowadays, nobody really de novo sequence. That's really what Don Hunt is known for. Now you have search algorithms, and the way that works, just to give you a very brief overview, and those in the audience who knows, I apologize for being, spending time on it, but you take a protein mixture, or your immune system takes a protein, you have a protein database, and you digest. You're predicting epitopes. We are calculating what the cleavage should be with trypsin. We can calculate, of course, the mass that this peptide should have. If this peptide can carry a post-translational modification, this peptide, of course, can be observed at either the unmodified mass, a modified mass, and if it had more than one modifiable residues, so on and so forth. So you can predict all this. You then look at the mass spectrum. You measure a peptide, this one right here. It's a doubly charged ion. So you pull out all the peptides that would fit in with the mass accuracy of your mass spectrometer to this mass. Then you record the MS2 spectrum. You record the theoretical fragmentation spectrum of these, and then you use different algorithm and compare which one is more likely. And of course, you have certain assumption in your algorithm that if these assumptions aren't true, your identification could mean anything, and that in HLA peptides is not, is often the case. So, and I hope I will show you throughout the talk what I mean by this. So, and what I would also like to point out, so I'm really jealous of the field of genomics, or maybe I'm not, because proteomics, it's difficult, it's a lot of fun for me to work in it, but when we look at the success rate of a proteomics experiment, if these are all the proteins or proteome forms in your cell, even with the most sophisticated mass spectrometry method, we're probably detecting about this amount of the proteins. The rest goes undetected. That goes back to the abundance bias. So what can one do to increase this range? You can fractionate your sample, which is a lot of people do. You just take your proteins, you separate them apart, so you give them in portions, so you get each one a little bit better. Or if you look for specific modifications like phosphorylation, you enrich for phosphorylated peptides. So, and the other problem with mass spectrometry is that not all peptides are equally detectable. So if you digest with trypsin, of course you will generate, depending how many basic residues, or you have enzymes that cleave after acidic residues, then of course you have the proteasome. If the peptide is too short, we won't see it. If it's too long, we won't see it. If it's too hydrophobic, we won't see it. So the nature of the peptide also dictates its detectability. So, and of course, if that peptide would carry a modification, you would also miss it because you don't see the peptide. So that goes back to really what the previous speaker said. So he was really right. So in this audience, I don't have to tell you about the progress of getting peptides onto the MHC molecules, but they're digested by the proteasome. This is the MH2 class pathway where we take endogenous proteins and put them out on the surface. And then, of course, the ideas that Don Hunt had very long ago, and I'm sure he was not the only one, but we always knew when we were students that in cancer, phosphorylation is upregulated, is dysregulated, so is glycosylation. So these modified peptides might be presented and be suddenly immunogenic. So, and what I would like to show you now is some historic slides from Don, from his paper in 1992, which was the first time that someone sequenced actually an HLA peptide. So what you see here is a mass spectrometer that back in the day was state-of-the-art. You can see the peak is rather broad. What you see here is the cell line. Let me just pull it up here. It's this cell line with this HLA on the surface. The next one is the control line that doesn't have this expressed. So you don't see anything in this region, but you see a peptide in this region. This is the amount of cells he used back then, which is not that far off what people are actually using nowadays. He purified this HLA allele using a specific antibody. That's pretty much what people still do today. He eluted with acetic acid, he boiled it off the MHC, and then he sent it actually to 5kD filter to just get the peptides. And then per analysis injection into the mass spec, if you don't assume any losses, he injected about an equivalent of 10 to the 8 cells. And then this is the very first spectrum. So in this spectrum, that was at a time there were no such algorithm. There were really not even, so this is all done by de novo sequencing. And isoleucine and leucine has the same mass, so we can't really differentiate which is which. And usually in mass spectrometry, you also don't observe cleavage in the first residue right here. So you couldn't even tell if it's an isoleucine-leucine followed by a threonine or a threonine followed by an isoleucine-leucine. So it was done by mass spec. Part of it was done by Edman degradation to really get the order of the first two residues. And back in the day, GenBank entry was less than 1% of what we currently have. So now you got a peptide, but you kind of had to figure out what protein it comes from, actually. So luckily, right around that time, the BLAST algorithm was published. So, and it turned out that this peptide came from this protein right here. So a couple of years, like at the same time, Don also worked on the MS splitter technology, where you take your HL peptide solution that I just showed you that you prepare by enriching these HLA molecules, and you take about 5/6 to go to the mass spectrometer to detect and get a mass reading, and 1/6 you spit into a 96-well plate that you use for a T-cell assay, which now I learned doesn't really mean much, but anyway, so you send it into the T-cell assay and you look for killing. If you find a fraction that has killing, you then go back to the mass spec and try to see what could cause the killing in this fraction. And this was done 2 years later in 1994, published with Vic Engelhardt in Science. And what you see here is, you probably can't see it, but this is a fraction that has activity. It's present in fraction 40, 41, and 42. And there's 3 peptides present here that are in all of these 3 fractions. And this is how a mass spectrum back then looked like. So you see a lot of noise here. But 3 peptides were measured in all of these fractions that had activity. They then tested it in 2 different melanoma cell lines, and these are it's hard to see, but there's 3 lines. So this is the line where there's no killing in a T-cell assay, but 1 peptide in 2 different melanoma cell lines has killing activity. So and that's how they back then, that's like over 25 years that they identified those. So, and of course I forgot to put down which melanoma specific peptide that was, but I think, this is by now. So this is, I don't know, you can probably not see that. This is just, I would like to give an overview of what people use to try to define these HLA peptides by mass spectrometry. You can, of course, use primary human cell lines, which this is the blue line. This gives you the best translational potential. But of course, it's a human cell line. You don't have much material. It's tedious to grow, and it's also hard to reproduce. You can use the mouse model, which of course gets you easier. So you get more, I can't even read that, reproducibility, availability. So it's a lot of easier, but of course the translational potential, again, from mouse to human is not as big. Or you can do a cell line, which of course you can easily grow. You get a lot of material, a lot of peptides to detect by mass spec, but the translational potential is a little bit hard. Then the next step, what people do is they either do immunopurification, which you need the most material about. You have the best selectivity, but reproducibility and available of material, of course, is harder because you lose a lot in the purification. You can then also wash peptides off the surface. That gives you a lot of material. But of course, the specificity and the reproducibility might not be as good, sorry. Or you can do strong acid elution. That's the same thing. So the mild acid elution, the idea is that you can wash them off the MHC molecule, let them grow, and they repopulate, and you can reassemble. And this one here, you just get everything that's on the surface off. And I think this is amount here. What is green? This is amount or like material, I guess. So you get the most material if you just acid wash your surface. And then, again, you won't really see that, but I will go through it in the next couple of slides. You then have, in principle, 3 ways of how you analyze this by mass spec. You can say, let's do discovery. We sequence, as we always sequence, the most abundant first, and then we narrow our way down. Or if you want to really prove if a specific peptide is present that might not be very abundant, you can set the mass spectrometer to just target that specific peptide. You will not see anything you have not anticipated before, but you have a little bit more sensitivity in potentially detecting the peptides you would like to go after. So how is it nowadays done? So I gave you a couple of historical slides out how it used to be done to get from a spectrum to a sequence. So nowadays we have search engine, and that's not even all search engines. We have Sequest, Mascot, Bionic, which is produced by my collaborators that are just 10 minutes down the road. Andromeda, which is from Matthias Mann, Protein Pilot, AB Science. So there's endless algorithm that translate this spectrum into a sequence. And nowadays, of course, we also have a reference proteome, and we also, thanks to next generation sequencing, we have personal proteomes. So if you have a tumor-specific patient and you want to know what, we usually have the proteome that we can search against. And even though we have all of this, roughly from all the spectra that we acquire, we have a success rate of getting the sequence that's about 10%. So, that might be debatable, and that might be, 2 or 3 years ago, but I want to just show you now, I kind of alluded through it in the introduction, but I want to show you why we might not get a match. And the first assumption is that the peptide is in the database. Well, nowadays, that's doable for most part due to exome sequencing, which the previous speaker already explained. And in this audience, I don't really have to. You have to tell me, but, and then you can, either take these peptides, check if they have recognition, predict algorithm, or identify these neoantigens by mass spectrometry. So exome sequencing really helps that we at least get the mutated sequences now that we can search. Before that, if there was a mutation and we don't have it in our database, we don't find it. But even though we have exome sequencing, these peptides might not necessarily still be in the database. And this is a paper also that the previous speaker mentioned that came out in Science in 2016, where they realized that there's a peptide that's a spliced peptide that's recognized in diabetes, and it's causing the killing of the beta cells. And that is a peptide that's comprised of the C-peptide of insulin. It's hard to see, but it's the first 6 or 7 residues are from the C-peptide of insulin. And the last 4 of this peptide are from a different protein that's found in these granules in the beta cells. So this peptide basically gets, if you break down a peptide, the reaction can always go back and forth. So you can break a peptide, but at the same time, you can also make a peptide backbone. You can do the reverse reaction. And that's what happens here. Well, if that person wasn't really looking hard and know how to interpret the spectra, he would have never found it. Because in just searching the data using search algorithm, this peptide would have never been found because it's fused by 2 different papers. And then just at the same year, also Albert Heck, the mass spectrometrist, came up with a paper that said that a large fraction of these class 1 ligands are proteasome-generated splice peptides. So that's a shockingly large number, and I'm not quite sure if I'm 100% agreeing with this. It's hard to imagine, but it's hard to see also, but the idea is that you have a protein, you cut out a middle region, you fuse them together, and now you suddenly have a new peptide. Again, if it's not in the database in mass spectrometry, and you just use a search algorithm, you will never see those peptides. At the same time, you can also cut it, and these peptides get back together in a reversed order. Again, something you cannot look for. And then there is some evidence, albeit not, not as much, but theoretically you could have also a fusion peptide of 2 different proteins, and that was shown by this group to actually be the case, because they really proved that this peptide comes out.
The problem is you're going to have so much immunogenic.
I know, that's why it's hard to believe, because we would all be, that's just my opinion, but you guys know this better than me. It's hard to believe. I would be terrified. I have an autoimmune disease just standing here right now. So on the second assumption, so I just told you the assumption is peptide is in the database, not always the case. The other assumption is that the MSMS spectrum, once we actually did acquire it, that it will give us a sequence, and that's not really always the case. What you see here is a sulfated peptide, so this was not isolated from an HLA molecule. I'm just putting it out as an example. So if you do the conventional fragmentation of CID, or now often also called HCD, all you see is the neutral loss of this modification, because the modification breaks much easier than the peptide bond, and you don't see any fragmentation. So I was a grad student in Don's lab when he developed ETD using anions to break peptides apart, and that is shown here. Oops, sorry. So usually we break the peptide bond apart right here, and in ETD, which is an anion analog of ECD that you might have heard for, that's electron capture dissociation, where we cleave is the amide nitrogen and the alpha carbon, and they're by definition called C and Z ions. So, and what's different between these two, on the top you put in little portions of energy. As soon as enough energy is reached for a peptide bond to break, the peptide bond breaks. So you have a modification that's very labile. What will happen? That modification will break, but not your peptide backbone. In ETD, you have a radical-driven mechanism, and it's not so much sequence dependent. It still depends on other things, but you get more random fragmentation. So, and the idea now that peptide that I showed you right now, that with conventional fragmentation don't give you a sequence, gives you a very nice fragmentation pattern using this fragmentation. And then the same thing for glycosylated peptide. This is the conventional way of fragmenting a glycosylated peptide. You don't observe much fragmentation. You subject it to ETD, and the more charged, the better. You see a very beautiful spectrum. You can see the peptide sequence, and you can see that it's glycosylated on this threonine residue. And then this is hard to see, but that's a slide that Don actually gave me. So his motivation, he's always, HLA peptides are his passion, and this ETD fragmentation was really the driving force, because lots of phosphopeptides lose the phosphate group, and we don't get the sequence. So now you can't really see it, but the red little dot, it's a phosphorylated serine or threonine. So Don applied this ETD then for the sequence of HLA peptides that are actually phosphorylated. And the idea is that in cancer, phosphorylation is out of order, and you get so many phosphorylated proteins that you usually don't have. And this peptide right here you can't see it, but it's this one right here. It's insulin receptor receptor substrate 2. Yep, thank you very much. So what you see here is the Western blot for the protein, and down here is for the phosphorylated residue that was found by mass spectrometry. And you can see that it's not necessarily the greatest Western blot, but you see it's present in this melanoma, a little bit here. This is a control cell line where you don't see a shadow. You see it in ovarian, and you can see it in some of these breast cancers. So, and then, it's even harder to read. And then where we also use ETD for is glycosylated peptides. So glycosylated peptide, this is actually a relatively good one. But what you usually see is, and you can't see it on this slide, but you see lots of white peaks that have no explanation, and they contain the sugar group. But as soon as we see this loss of sugar, this has a very diagnostic ion, we then trigger an ETD on the same precursor, and then we can get the sequence. And more importantly, we can get the exact location of the sugar group on this peptide using this different fragmentation. And Don just recently published a paper where he found 93 different glycopeptides from 3 different melanoma cell lines. Again, these are cell lines, but this is the first time he found so many. Well, Albert Hecht might have too, so I better be careful when I say who did what first. But the glycosylation was definitely diverse, 17 different glycoforms, and that's only, a bucket. There were so many spectra that were still not interpretable that clearly carried a sugar, but the sequence was not detectable. And they even found disulfide-bonded peptides, glycopeptides, that are represented here in these These are class 2 peptides. So with this, I would like to just, emphasize again, like certain peptides, like this one here, fragments beautiful with CID, doesn't give us any fragmentation info with ETD. This peptide here in CID doesn't give us good fragmentation, but gives us beautiful fragmentation in ETD. But then there are unfortunately also a large percentage of peptide that don't really fragment in either way. And if we can't fragment and can't get a sequence, we can't say if this is a potential active neoepitope or not. So, and that's when Albert Hecht, about 3 years ago, came up with yet another fragmentation that combines ETD with HCD, and he calls it ETHCD. So he fragments the peptide first using ETD, and then takes all the fragment ions together and activate them with HCD, and then you actually see both fragmentation ions. And this is an HLA peptide where you get decent coverage with ETD, not so good with HCD, but if you put this fragmentation technique, you can get decent coverage. And ideally, you want to see every single peptide backbone cleavage so you can really be sure what the sequence is. And when he did this, he got a 3-fold increase in the HLA peptide identification. So the next assumption that you hope you have when you detect HLA peptides by mass spec is that you actually select them for MS-MS. And so this is a full scan spectrum, and usually what we do, we take the 10 most abundant ions, individually isolate them, fragment them, then we acquire another full scan, and that usually is less than 3 seconds later, so the spectrum hasn't really changed much. And whatever we just already isolate is on an exclusion list, and we go to the next most abundant one. But that seems a lot, and we're getting faster. Now we can do top 40. When I started, we were able to do maybe top 5 in 3 seconds. But it's usually not enough. So what you can do is you can do a targeted analysis. You don't care what is the most abundant one. You just set the mass spectrometer to just give you this peptide or this specific mass. You don't even have to see it. You can just set the mass filter to just select the ion of interest throughout the entire run. And this is supposedly clearly illustrated here, but I don't think you can see it very well. These are two different types of mass spectrometers. The triple quads, if you're familiar with them, are traditionally used for targeted MRM assays, but you have to know a priori what fragment ions you want to monitor to see it, and then you can very accurately determine it. The new generation Orbitraps, you don't have to know necessarily what fragment ions you want to assay. You can do this later on, but you do have to, just in the first one, have a peak list of masses that you would like to assay for, and then you just look for those masses in the mass spectrometer. In this case, every colored ball is a different peptide of a different mass, and you only want to see the green one, so you only let the green one actually into the mass spectrometer. And then this is an example of targeted analysis that I did with Ralph Steinman's lab back in the day in Rockefeller. So this is old data where we tried to take the GAG P24 protein, pulse it onto dendritic cells, and then hopefully find the protein back onto the surface molecule. So what we did is my collaborator Leonia made the dendritic cell lines. We took the GAG24 protein and we calculated all the possible MHC2 peptides there could be without any motif. We just predicted masses. We run the LCMS, and then what we do, we look for these masses. So what you see on the left side is the theoretical mass pattern of one of those peptides. So in black, which you can barely see, is the mass of the peptide. In red is carrying one carbon-13 isotope, and in green, the second carbon-13 isotope. So in mass spectrometry, we don't see one peak, we see a peak with isotope patterns. And when we know the sequence, we can actually predict what the ratio of isotope patterns should be. So what you see on the right side is a peptide. It's very noisy, it's noisy data. And there is a mass that really fits the right mass, but the isotope ratio is not the right thing. So we exclude this. But here was a peptide that has a mass that is dead on, and an isotope ratio that seems reasonably good. But this is, it's noisy because it's very, very, very low abundant. So what we then did after we saw this, we checked at what retention time we saw this, we targeted for this peptide, and then we could confirm that it really is indeed the GAG P24 peptide from this protein that we saw. So it takes a lot of time and effort to really find those. And even though they pulsed them with those, these were really extremely low in the grass that we could identify them. So, and then this is a recent paper. Again, I think this is by the Heck group in 2016. So they started with stimulated dendritic cell lines. And again, this is close to the amount that Don Hunt used about 25 years ago. They pulse them, they take the proteasome of this cell line, so they take the proteins, they isolate the HLA class 2 DR using an antibody, they get the peptides, they take these peptides and they fractionate them into 10 different fractions for dynamic range reasons that I mentioned in the very beginning, and then they acquired mass spec. They have about 30 LCMS runs where they analyze those, and they were able to find 14,000 unique peptides from under 2,000 source proteins. And what I would like to point out on the peptides that they identified, they acquired each sample with either ETHCD, ETD, or just HCD. And the overlap, you can't read it, but it's just 20% of the peptides were found in all of these analyses. So you see that mass spectrometry really, the previous speaker was right, it's not universally able to detect everything. Every fragmentation method really has a sweet spot of what you can use. So and here they used all three to get this number of peptides detected. So, and this is that's also the previous speaker mentioned this. This is a paper by Matthias Mann and Michel Bassani-Sternberg. That's in 2016, where, I don't know if you can see, but they took a tumor from a patient. They isolated the MHC molecules using an antibody and then did mass spec. And at the same time, they subjected the tumor to whole exome sequencing, got the mutation, so they have a database that they then can search the data against. And then I can't even read this, but then they do some testing afterwards if these peptides that they found really are immunogenic, I believe. So, and what they used is, depending on what, they had several tumors they looked at, they used 0.1 to 4 gram of tumor tissue They IP'd these peptides using either pan-HLA1 or HLA2 antibodies. They had 25 different melanoma samples. This was 140 different LC-MS runs. And they found about 80,000 class 1 peptide sequences, assuming a false discovery rate of 1%. So again, which all I told you just how difficult it is to interpret or misinterpret or miss something, so take this for whatever you want. And they found 365 phosphopeptides, and a third has never been reported in the literature. And that's probably the literature that's reported is all tryptic peptides. So here we have peptides that might have never been generated from a proteome before, and that's why we see them. And they also detected 11 of all of these 80,000, they detected 11 of these neoepitopes. So 80,000 versus 11. So it's, you know, so, and just in a summary to end with this talk, and I think that's where I'm going to talk the topic because it's getting late. But usually what people need is still a 5 to 10 to the 8 cells with that many MHC molecules expressing. So this is still not that different from what Don used about 25 years ago. But what helps really now to get, you know, to have a better sequencing of these peptides is the different MS fragmentation method. Exome sequencing, of course, helps. And the search algorithms also get much more sophisticated in helping interpreting and not making false assumptions in interpreting this data. And that's how people come up with these numbers of 1,000 HLA peptides identified. And the second part of the talk is what I'm working on antibody sequencing, but I'm just going to skip over this because it's getting late. So, but we sequenced antibodies of patients that have that HIV infected or have malaria, and we can characterize their antibodies. Oops. And I just want to point out my colleagues, they write the software of sequencing HLA peptides and also these antibody peptides that are not trivial and not the usual proteomics data set. And they're just down on, in San Carlos, so they're just 10 minutes away from here. And sorry. Okay, so and I'm going to thank my lab, of course. And Avantika is working with MedGenome, so she's right here. And of course, I thank Don Hunt very much and Jeff Shabanovitz, also especially Don for sending me slides. And then, of course, my collaborators, who I couldn't really talk about how, you know, the project I'm working on with them. And then I didn't mention these talks right now. But thank you very much for your attention. Thank you.